start with our session today. Okay, so we'll be talking about the introduction about the endocrine physiology. Okay, so here in the image you can see there are several endocrine glands throughout our body located at different parts. For example, in our hypothalamus region, below that you can see there is a pituitary gland, and also beside that you can see there is a pineal gland. Okay, then in our thoracic region we have a thyroid gland, and there we have also parathyroid gland. Then we have a thymus gland. Okay, and in our digestive system also possess several endocrine glands, where in the pancreas also we have those islets of Langerhans that will also produce hormones. Okay, and about the kidney we have the adrenal glands. Okay, and also our ovaries, and in male the testes. There are also several endocrine hormones are being secreted in our body that regulate the functions of our body. Okay, so here you can see there is a chemical signals act over short and long distances. Okay, so as there is a neuron, we know that that also secretes some sort of signals, some uh, sort of, you can say, chemicals. Okay, that is also being secreted. For example, here you can see this is a neuron, and at the end of the neuron you have a synaptic vesicles, and that release those chemicals, like fuse with the membrane and release that chemicals in the synaptic cleft, and in the postsynaptic cells having the receptor for that, and that will carry out the message to the next neurons. Okay, here we can see a non-neural endocrine cell. Okay, so there is a cell that is also with the same method. These vesicles fuse with the membrane and also releases the hormones. Okay, and that goes into the circulation, so the capillary near the secreting cells will receive those hormones, and inside these capillaries these hormones travels. Okay, so here you can see there are larger blood vessels, then from the capillaries goes to the larger blood vessels, and then from the larger blood vessels it follows the capillary near the target cell. So one which is a secreting cell, hormone secreting cell, and at another position we have a receptor cells, okay, or target cell which harbors the receptor for those hormones. So via the blood these hormones travel and goes to the target cells, where it binds to their respective receptors and act on those particular cells. Okay, and also one more thing which is very crucial here: in case of hormones you need very small quantity of hormone to carry out the physiological functions compared to the neuronal signals of neuronal chemicals. Okay, here also you can, it is a neurosecretory cell. Okay, so this is a capillary near secreting cell terminal, the blood vessels, and then yeah, so here it is a non-neural endocrine cell. This is a neuron which is secreting the hormones and which is carrying with the bloodstream and going to the target cells. Here you can see, so this is also one more difference, which is a paracrine versus autocrine. So if there is a one cell which is secreting the hormone, secretory cell, another cell which is a target cell, and their hormone is acting, that is called a paracrine system. And if the same cell which is secreting the hormone also having the receptor, so the hormone which is being secreted also like targeting that cell itself, so that is called an autocrine signal system. Okay, so this is a if we talk about the chemical structure, the nature of the hormones. Okay, so there are three general classes of hormones. So all the hormones, like there are majority of the hormones are protein and polypeptides, so including hormones secreted by the anterior and posterior pituitary glands. Okay, from the pancreas the insulin and glucagon hormones are being releases, and the parathyroid gland releases the parathyroid hormones, and many other majority hormones are the protein or polypeptides. Then we have the steroids hormone. Okay, that's secreted by the adrenal cortex. Okay, for example cortisol or aldosterone. Ovaries also secrete estrogen and progesterone, and testes secretes the testosterone, and the placenta secretes the estrogen and progesterone. So these are all the steroid hormones. And also there are certain derivatives of amino acid tyrosine. Okay, for example, which is secreted by the thyroid cells. So thyroxine and triiodothyronine, and the adrenal medulla hormone epinephrine, norepinephrine, and these are, there are no known polysaccharide or no nucleic acid hormones. Okay, so this is a derivative of tyrosine, these hormones. Okay, so here you can see tyrosine derivative hormones that includes epinephrine, norepinephrine, dopamine, thyroxine, triiodothyronine, steroids. We have the testosterone, estradiol, progesterone, cortisol, aldosterone, vitamin D. We have several peptide hormones: oxytocin, vasopressin, angiotensin, melanocyte stimulating hormone. Okay, the somatostatin hormone. Okay, so somatostatin also we have discussed in the last class. Okay, somatotropic inhibiting hormone that is having the target cell of parietal cell, which is secreted by the D cell inside the stomach. Okay, then we have the thyrotropin releasing hormone, gonadotropin releasing hormone. We have the protein hormones. Majority are the protein hormones, including insulin, glucagon, adrenocorticotropic hormone, thyroid stimulating hormone, luteinizing hormone. Okay, calcitonin, parathyroid. All these are the protein hormone, which is having a length of more than 20 amino acid. If the length is less than 20 amino acid, these are called as peptide hormones. Okay, so here how these hormones are targeted to the cell. So this is a target cell. So if it is a suppose steroid hormone, it can easily cross the plasma membrane. It has a target inside the nucleus. Okay, it goes and bind to the receptor. The receptor is present inside the nucleus, and that receptor binds to the DNA and activates the transcription factor and allow the synthesis of the target gene under the influence of that hormone. Okay, so certain hormones which are like, here you can say polypeptide hormones, so they are having receptor. So here you can see this is a G coupled uh, G protein coupled receptor, which is producing the cyclic AMP, which is secondary messenger, and then several kinases which activates the transcription factor and allow the synthesis of the target genes and the proteins that will result from that, and that will cause the biological response. And the other mechanism is so the tyrosine hormones. Okay, so there is this, for example, this IGF, the growth factor. So it binds to the receptor that ultimately phosphorylates the tyrosine, and that will further activate the transcription factor, and same way biological response will happen. Okay, so these are the hormones in the blood. So hormones are secreted into extracellular fluid and readily enter the blood by passive diffusion driven by steep concentration gradients. Okay, so diffusion through the pores in capillary endothelium. Okay, so these are secreted into extracellular fluid. Okay, and diffusion through the pores in the capillary endothelium also largely accounts for delivery of hormone to the extracellular fluid that binds the both target and non-target cells. So receptor mediated transfer across capillary endothelial cell may facilitate delivery of insulin and perhaps other hormones to target cells, but the importance of this mechanism of hormone delivery has not been established. So, in general, hormones distribute rapidly throughout the extracellular fluid and are not preferentially directed towards their target tissue. Okay, and here you can see these are the half-life described by of the hormone. So here you can see amines are having 2-3 minutes lifetime half-life. Thyroid hormones are having 6-7 days of half-life. Polypeptides having 4 to 40 minutes. Okay, steroids have 4 to 120 minutes half-life. Okay, so that varies. So the half-life of a hormone in blood is defined as that of the period of time needed for its concentration to be reduced by half. Okay, so the clearance of hormone from blood depends on the rapidity with which it can escape from the circulation to equilibrate with extravascular fluids, as well as on its rate of degradation. So some hormones, for example epinephrine, have half-lives of the order of seconds, whereas thyroid hormone have half-life in order of days. So the half-life of a hormone in blood must be distinguished from the duration of its hormonal effect. So some hormones produce effects virtually instantaneously, and the effect may disappear as rapidly as the hormone is cleared from the blood. Other hormones produce effects only after a lag time that may last minutes or even hours. So, and the time the maximum effect is seen may bear little relation to the time of the maximum hormone concentration in the blood. So let's understand. So there are some hormones which binds to the protein. So most peptide hormones are quite soluble and they circulate completely dissolved in the plasma water. Okay, so some peptide hormones, they dissolve into the plasma and then they circulate. And then we have the steroid hormones and thyroid hormones whose solubility in water is limited and circulate bound specifically to large carrier proteins with only a small fraction, sometimes less than 10%, okay, present in free solution. So some peptide hormones also bind to specific protein in plasma, so which is protein binding, and this protein binding is reversible. Free and bound hormone are in equilibrium. So however, only free hormones can cross the capillary endothelial and reach its receptor in target cells. Okay, so protein binding protects against loss of hormone by the kidney, slow the rates of hormone degradation by decreasing cellular uptake, and buffers changes in the free hormone concentration. So here you can see, so this is a hormone thyroxine. So the bound hormone represents a reservoir of hormone and as such can serve to buffer acute changes in hormone secretion. So the hormone remains bind to this protein and whenever it's needed it can be released. Okay, and it serve as a buffer. As the concentration changes, the bound hormone is released from the protein. Okay, here you can see, so it is a carrier bound hormone which can be interconvertibly changed into free hormone. Okay, it can also, it's a free hormone can lead to the hormone degradation, that's why for the protection of hormone also it is carrier bound. Okay, endocrine cell also releases some free hormones, later it can bind to the carrier protein and this free hormone can bind to the hormone receptor in order to generate the biological effects. Okay, so hormone degradation also we can study. Okay, so degradation of hormone, their subsequent excretion as processes that are just as important as secretion. So in general, the rates of hormone degradation are characteristic for each hormone and follow first order kinetics. That is, a constant percentage of hormone present in blood is destroyed per unit time by processes that are not usually subject to regulation. Okay, so inactivation may involve complete metabolism so that no recognizable product appears in urine, or it may be limited to some simple one or two step process such as addition of a methyl group or glucuronic acid. Here you can say the most insulin molecules are degraded by liver cells with the help of enzyme insulinase. Okay, so here you can say endocrine cell, the hormone which is there in the capillary, and this is the target cell. So in the liver, phase 1 reduced and hydroxyl, phase 2 conjugated. Okay, so there the enzyme which is present, which is for the degradation of this hormone, is insulinase. Okay, and there is a negative feedback. So secretion of most hormones is regulated by negative feedback. Okay, so negative feedback means that some of the consequence of hormone secretion acts directly or indirectly on the secretory cells in a negative way to inhibit further secretion. Okay, so if there is a lot of concentration of that hormone, it will further give the inhibitory signal. Okay, so yeah, so here you can see like, for example, this is the alpha cell which secretes the glucagon. Okay, and when the glucagon is secreted, liver is making the glucose converted to glycogen and storing it. That causes the reduction in the uh, glucose concentration, and that gives a feedback to the alpha cell for the lower production of the glucagon. Okay, so a fall in blood glucose detected by the alpha cell of the islets of Langerhans causes them to release the glucagon, which stimulate the liver to release glucose and uh, thereby increase blood glucose concentration. Yeah, sorry, yeah, so it was the opposite effect. So glucagon of the effect of glucagon on the liver is to increase the glucose concentration, to break the glycogen to glucose. So with restoration of blood glucose to the set point level, further secretion of glucagon is inhibited, and this simple example involves only secreting cells and responding cells. Okay, so for example here, so there are two basic configuration of negative feedback loops within the endocrine system. So a physiological response driven feedback loop referred as response driven feedback and an endocrine axis driven feedback loop. So these are the two feedback loops. So physiological response driven negative feedback. So this is an endocrine gland which is releasing this hormone which is working on the target organs and showing the physiological effects. Okay, for example, the glucagon one, the simple one. So after the physiological effects shown, for example, blood glucose. So when the blood glucose increases, then it again gives the signal to endocrine gland to reduce the secretion of this hormone, so that further processes will also slow down. This is a physiological response driven negative feedback. Then this is the endocrine axis driven negative feedback. So for example, so this is the hypothalamic neuroendocrine um, neurons. Okay, so the neurons, they release certain hormone that will act on the pituitary gland. Okay, so pituitary gland releases the trophic hormone. Okay, and this trophic hormone act on the peripheral endocrine glands. For example, it act on the adrenal gland or some other glands. So the pituitary gland releases some trophic hormone, and those trophic hormone act on the endocrine glands, peripheral endocrine gland, and that will release the main hormone. So when this main hormone so then this hormone's concentration increases, that will work on the target organs, and then it will be having the physiological effects. So when the hormone and then this hormone also give the negative feedback to the pituitary gland and also the hypothalamic neuroendocrine neurons. Okay, so they will slow down the secretion or stop the secretion of releasing hormone. Also the pituitary will slow down the secretion of trophic hormone. So ultimately there will be a lower secretion of peripheral endocrine gland hormone release. Okay, so let's talk about the growth hormone. This is the development of pituitary gland. Okay, so here you can see it is a pituitary gland has two parts. This is the posterior pituitary and this is the anterior pituitary. Okay, here you can see their anterior pituitary hormones. So TSH, which will work on thyroid gland, and thyroid gland will release the thyroid hormones T3 T4, and other which is ACTH, which will act on the adrenal cortex and that will release the glucocorticoids, for example cortico. Okay, I'll zoom it a little. Okay, so thyroid stimulating hormone, you can say. So it is allowing acting on thyroid gland, releasing the T3 T4 hormones that will affect the metabolism and the growth. This um, ACTH hormone, it will act on the adrenal cortex, which will release the glucocorticoids, which is having a role in stress response and metabolic actions. Then pituitary also releases the growth hormone, okay, which will have a positive impact on liver. So so this will release IGF, which is insulin like growth factors for growth of many tissues and also another tissues, and that will provide the metabolic actions. Then it also releases the luteinizing hormone and follicle stimulating hormone that will have the positive impact on gonads, ovaries in female and testes in male. So it will be having a role in sex hormone production and secretion, and sperm production in males, follicle development and secretion in females. Okay, so here this is MSH in some animals. Okay, and it is having a function in skin darkening in amphibians and reptiles except birds and fish, and prolactin also, which is having the positive impacts on mammary glands in mammals and various vertebrates. Okay, and here you can see this is the posterior. So these are the anterior pituitary hormones. So from anterior pituitary, so neurosecretory cells in the hypothalamus, they secrete the neurohormones at the capillaries of the median eminence. Okay, and then here you can see, so which travels through the portal vessel to the anterior pituitary and stimulates or inhibits secretion by the anterior pituitary endocrine cells. And here these are the neurosecretory cells in the hypothalamus, secretes oxytocin and vasopressin at the capillary in the parts of pars nervosa. Yeah, so these are the cells and hormones of the anterior pituitary we just now saw. So somatotropes are there, which releases the growth hormones, corticotropes, which are the this ACTH, thyrotropes and gonadotropes, which is luteinizing hormone and follicle stimulating hormones, and this prolactin. Okay, so growth hormones, let's talk about growth hormones. So it hormone promotes growth of many body tissues. So it is also called somatotropic hormone, or somatotropin is a small protein molecule that contains 191 amino acid in a single chain and has a molecular weight of 22.05. So although the anterior pituitary gland produces at least 6 hormones, more than one third of its cells synthesize and secrete gonadotropic hormone. Growth hormone. So it causes growth of almost all tissue of the body that are capable of growing. So it promotes increased sizes of the cell and increased mitosis with development of greater number of cells and specific differentiation of certain type of cells such as bone growth cells and early muscle cells. So this is the growth hormone. Okay, so this figure shows marked enhancement of the growth in the rat given growth hormone. Okay, so in the early days of the life and even after the 2 days, two rats reached adulthood. In the early stages of the development, all organs of the treated rat increased proportionately in size. After adulthood was reached, most of the bones stopped lengthening, but many of the soft tissue continued to grow. So this result from the fact that once the epiphyses of the long bones have united with the shaft, further lengthening of bone cannot occur, even though most of other tissue of the body can continue to grow throughout the life. So this is a growth hormone binding protein composed of 237 amino acid. Okay, so about half of the growth hormone in blood circulates bound to a protein, so that has the same amino acid sequence as the extracellular domain of growth hormone. So in fact, the plasma growth hormone binding protein is a product of the same gene that encodes growth hormone receptor and originates by proteolytic cleavage of the receptor at the outer surface of the target cell. Okay, so the same thing, just a cleaved portion of the receptor of the growth hormone. So it is thought that the binding protein serves as a reservoir of growth hormone that buffers the changes in the free hormone concentration and prolongs the half-life of the hormone. So the monomeric form of the growth hormone can readily cross capillary membranes, but bound membranes presumably cannot. Okay, so the half-life of this hormone is about 20 minutes, and that crosses the glomerular membrane, is reabsorbed and destroyed in the kidney, so which is the major site of growth hormone degradation. So less than 0.01% of the hormone secreted each day reaches the urine in unrecognizable form. Okay, and growth hormone is also degraded in its various target cell following uptake by receptor mediated endocytosis. Okay, so this is the mode of action for the growth hormone. This is a growth hormone bound to the receptor. Okay, so like other peptide and protein hormones, growth hormone binds to its receptor on the surface of target cell. Okay, on the surface of target cell. So the growth hormone receptor is a glycoprotein that has a single membrane spanning region and a relatively long intracellular tail. It neither has catalytic activity nor interacts with G proteins. So this growth hormone receptor binds to cytosolic enzyme called Janus kinase or JAK2, which catalyzes the phosphorylation of the receptor. Okay, phosphorylation of receptor and other proteins on the tyrosine residues. Okay, so growth hormone activates a signaling cascade by binding sequentially to two growth hormone receptor molecule to form a receptor dimer. Okay, form a receptor dimer that sandwiches the hormone between the two receptor molecules. Here you can see. Okay, so growth hormone has several metabolic effects. So aside from its general effect in causing growth, growth hormone has multiple specific metabolic effects. So for including increased rate of protein synthesis, okay, increased metabolization of fatty acid, decreased rate of glucose utilization. Growth hormone promotes protein deposition in tissues and also increased nuclear transport of DNA to form RNA. Okay, over more prolonged periods, growth hormone also stimulates the transcription of DNA in the nucleus, causing the formation of increased quantities of RNA. So, this promotes more protein synthesis and promotes growth if sufficient energy, amino acid, vitamins, and other requisites for growth are available. In the long run, this may be the most important function of growth hormone. So, decreased catabolism of protein and amino acid in order to uh, make a stock of the protein. Okay, so growth hormone enhances almost all facets of amino acids uptake and protein synthesis by the cell, while at the same time reducing the breakdown of the proteins. Okay, so growth hormone enhances fat utilization for energy. Okay, so growth hormone has a specific effect in causing the release of fatty acids from adipose tissue, and therefore increasing the concentration of fatty acid in the body fluid. Okay, so in addition, tissues throughout the body, growth hormone enhances the conversion of fatty acids to acetyl coenzyme A and its subsequent utilization for energy. So therefore, under the influence of growth hormone, fat is used for the energy in preference to the use of carbohydrates and proteins. Okay, so this is a ketogenic effect of growth hormone. So under the influence of excessive amounts of growth hormone, fat mobilization from adipose tissue sometimes becomes so great that the large quantities of acetoacetic acid are formed by the liver and released into the body fluids, thus causing ketosis. So the excessive mobilization of fat from the adipose tissue also frequently causes a fatty liver. Okay, so growth hormone decreases carbohydrate utilization. So we are talking that it increases fat utilization instead of carbohydrate as a source of energy. It increases the protein synthesis and lowers the protein degradation, and also it decreases the carbohydrate utilization. So it decreases the glucose uptake in the tissue such as skeletal muscle and fat. Okay, and increased glucose production by the liver and increased insulin secretion. So each of these changes result from growth hormone induced insulin resistance. Okay, so which attenuates insulin's action to stimulate the uptake and utilization of glucose in skeletal muscle and fat, and to inhibit gluconeogenesis. So this leads to increased blood glucose concentration and a compensatory increase in insulin secretion. So for these reasons, growth hormones effect are called diabetogenic. And excess secretion of growth hormone can produce metabolic disturbance very similar to those found in the patient with type 2 non-insulin dependent diabetes, who are very resistant to metabolic effects of insulin. Okay, so here this is a hypothalamic GHRH, and this is GHIH. So this is a releasing hormone and this is a inhibiting hormone. They both work on the anterior pituitary that increases the growth hormone secretion. Okay, so growth hormone works on liver, many other tissue, adipose tissue, and muscle. So let's see. So on liver, so it increases insulin like growth factor secretion that will work. So IGF, the growth factor, so it will work on many tissue that increases the cell division and other growth promoting events. Okay, then this is also the physiological response. So increases the protein synthesis and insulin like growth factor secretion. In adipose tissue, it decreases glucose uptake and increases the lipolysis for the lipid, um, for the fat to break for being utilized as an energy source. And in muscle also it decreases the glucose uptake and increase the amino acid uptake. Okay, so it altogether provides energy and substrates for growth. Okay, then we have the negative feedback. So long loop of negative feedback. So this IGF is having a negative feedback on anterior pituitary and the hypothalamus for the to stop the secretion of growth hormone. And also this growth hormone itself has a short loop negative feedback on the hypothalamus to uh, maybe inhibit the secretion of GHRH and increase the secretion of GHIH. Okay, so this is growth hormone stimulates cartilage and bone growth. So its most obvious effect to increase the growth of the skeletal frame. So what happens? So it increases deposition of protein by the chondrocytic and osteogenic cells that causes bone growth, and it increases rate of production of these cells, and specific effect of converting chondrocyte into osteogenic cells, thus causing deposition of new bone. Okay. So there are two principal mechanism of bone growth. So first, in response to growth hormone stimulation, so the long bone grows in length at the epiphyseal cartilage here. Okay, epiphyseal cartilage. Okay, so where the epiphyses at the ends of the bones are separated from the shaft. Okay, and this growth first causes deposition of new cartilage, okay, and followed by its conversion into new bone. Okay, so thus elongating the shaft and pushing the epiphysis farther and farther apart. At the same time, the epiphyseal cartilage itself is progressively used up, so that the late adolescence no additional epiphyseal cartilage remains to provide for further long bone growth. So at this time, bony fusion occurs between the shaft and the epiphysis at each end, so that no further lengthening of the long bone can occur. So here, so within the bony tissues, okay, so osteoblasts and osteoclasts perform their activities in to build, degrade, and thus constantly remodel bone. Okay, so osteoclasts begin degrading bones, and the resorption pits are partly filled by the new bone matrix produced by osteoblast, which is subsequently mineralized. Okay, here you can see. So osteoblast in the bone peristenium and in the some bone cavity deposit new bone on the surface of the older bone. Simultaneously, osteoclast in the bone remove the old bone. So when the rate of deposition is greater than of resorption, the thickness of the bone increases. So growth hormone strongly stimulates osteoblasts. Therefore, the bones can continue to become thicker and throughout the life under the influence of growth hormone. For instance, the jaw bones can be stimulated to grow even after adolescence, causing forward protrusion of the chin and the lower teeth. Likewise, the bones of the skull can grow in the thickness and give rise to the bony protrusion over the ears. So here is the growth hormone exerts much of its effect through intermediate substance called somatomedians, also called insulin-like growth factors. Okay, so this we know that growth hormone acts on the liver and releases the insulin-like growth factor 1. Okay, so when the growth hormone is supplied directly to cartilage, chondrocytes cultured outside the body. Okay, proliferation or enlargement of the chondrocytes usually fails to occur. Yet, growth hormone injected into the intact animal does cause proliferation and growth of the same cells. Okay, so in brief, it has been found that the growth hormone causes the liver and to much less extent other tissue to form several small proteins called somatomedians that have the potent effect of increasing all aspects of bone growth. So many of the somatomedians effect on the growth are similar to the effect of insulin on growth. So therefore the somatomedians are also called insulin like growth factor. Okay, so here you can see it's a growth hormone directly given. It is not having a effect on the growth of the bone, but it has an effect on the liver that will produce the IGF-1, and this is a bone largening is happening, so in presence of the insulin like growth factor 1. And here the it is what it is doing, growth hormone direct role is to metabolize the fat and break down the fat or do the lipolysis so that the fat can be utilized for as a energy source. So that is the direct effect of the growth hormone, and bone lengthening in via IGF-1 is the indirect effect of the growth hormone. Okay, so okay, so the pygmies of Africa have a congenital ability to synthesize significant amount of somatomedian C. So therefore, even though their plasma concentration of growth hormone is either normal or high, they have diminished amount of somatomedians in the plasma. This apparently accounts for the small stature of these people. Some other dwarfs also have this problem. So Laren syndrome or Larentyme dwarfism is an autosomal recessive disorder characterized by an insensitivity of growth hormone caused by the variant of growth hormone receptor. It causes short stature and resistance to diabetes and cancer. Here from the pituitary, growth hormone is being released and having an effect of bone and other tissue via the IGF-1. Okay, this IGF-1 further is also having an inhibitory effect. So the role of growth hormone, insulin growth factor, in promoting growth. So growth hormone stimulates IGF-1 production, okay, in liver and epiphyseal growth plates. So epiphyseal growth is stimulated primarily by autocrine, okay, autocrine or paracrine action of IGF-1. So autocrine action is the same cell which is producing the hormone having the target also. Okay, so the IGF-1 is showing a autocrine system. So hepatic production of IGF-1 stimulates circumferential growth of the bone and acts primarily as a negative feedback regulator of growth hormone. So liver is a principal source of IGF-1 in blood, but other growth hormone target organs may also contribute to small amount of circulating pool. So the growth hormone direct effect is on fat and carbohydrate, increases the lipolysis and increases the blood sugar as it slows down the uptake of the glucose. And the indirect effects are it targets the liver and other organs for the release of IGF-1. So from skeletal, what happens, chondrogenesis increases, and in muscle and adipose tissue, protein synthesis and cell proliferation increases. So these are the effect of the growth hormones. Okay. So regulation of growth hormone secretion. So in the hypothalamus, the PVN, periventricular nuclei, and arcuate nuclei, SST somatostatin. This is growth hormone releasing hormone. Okay. Yeah. So these are the hormones. So the growth hormone releasing hormone, and this is the PVN, which is the somatostatin. So it is a growth hormone releasing hormone, stimulates pituitary gland to release the growth hormone that will have an effect, positive effect on the liver, and that will produce IGF-1. IGF further inhibits the pituitary gland to inhibit the secretion of the growth hormone, and also it has the effect on this arcuate nuclei to inhibit the secretion of growth hormone releasing hormone. Okay, growth hormone can also directly inhibit the this PVN, periventricular nuclei, okay, to inhibit the secretion of the somatostatin growth hormone. So somatostatin hormone to increase the secretion of growth hormone. So this is a regulation of growth hormone in secreting cell. So the somatotrophs expresses cell surface receptor. Okay, so what is a somatostatin? We show you here. From the periventricular nucleus, somatostatin is released and inhibits the pituitary somatotrophs. Okay, and this is a growth hormone releasing hormone. This is somatostatin. Okay, and here what we are seeing, the somatotroph expresses cell surface receptors for growth hormone releasing hormone, 7 transmembrane domain G protein coupled receptor. Okay, so here this pituitary is a somatotroph. Okay, it is a somatotroph. Here I will be showing the effect of both of these hormones. Okay, so it has a receptor for growth hormone releasing hormone and also for somatostatin. Okay, so um, G protein coupled receptor. So ligand binding induces the conformational change that result in dissociation of the subunit from stimulatory G protein. Okay, and which activates the adenyl cyclase. Okay, here adenyl cyclase to generate cyclic AMP from ATP. Okay, from ATP to cyclic AMP. It is under the effect of growth hormone releasing hormone. Okay, so in turn activates the intracellular protein kinase. Okay, protein kinase here, inducing the protein kinase A, which phosphorylates and activates nuclear localization of CREB. here. Phosphorylates the CREB. So in the nucleus, CREB activates the transcription factor FOS. Okay, so FOS and transcription factor PIT1. Okay, FOS and PIT1 also activates the transcription of the growth hormone, this gene, growth hormone gene, so which promotes proliferation. So growth hormone synthesis and secretion are inhibited by the somatostatin. So at one hand you are saying that growth hormone releasing hormone is ultimately increasing the synthesis of growth hormone. Okay, and on the other hand, the growth hormone synthesis and secretion inhibited by the somatostatin, so which signal through the its G protein coupled receptor to activate the inhibitory GI protein. So GI signaling leads to reduced intracellular calcium levels and inhibition of adenylyl cyclase. Okay, so this is the basic concept. So one end, the growth hormone releasing hormone is promoting the transcription of growth hormone inside this somatotroph, so that the growth hormone is more growth hormone is being produced and released. Okay, and that helps in the proliferation and growth. And when the somatotroph is coming via this receptor, what it is doing, it is decreasing the calcium concentration, which affects the adenylyl cyclase. So ultimately it will not produce the cyclic AMP and all the downstream processes will be inhibited. Ultimately growth hormone synthesis is also being inhibited under the presence of somatostatin. This is acromegaly. So if there is a tumor in the growth hormone cells after adolescence, so that is after the epiphyses of the long bones have fused with the shaft, the person cannot grow taller, but the bones can become thicker and the soft tissue can continue to grow. Okay, this we know. So this condition is known as acromegaly. So enlargement is especially marked in the bones of the hands and the feet, in the cranium, nose, bosses of the forehead and supraorbital ridges and lower jaw bones and portion of the vertebrate, because their growth does not ceases at adolescence. Okay, so this is a condition. So yeah, so this is the end of the our topic for today. So let's see what the question that we have for today's class. So this is a question that we discussed in the last class. Okay, so last class I have discussed how the HCl is being secreted from the parietal cells. Okay, so what is there, we talked about the physiology of the digestive system. So from our mouth we have the esophagus. The esophagus go down and enters into the our stomach. Okay, in stomach also we have five parts. Our stomach is divided, and in one of the part we have these cells which are called as parietal cells, which produces the HCl. Okay, that helps in churning of the food. Okay, or digestion of the food. So this HCl is secreted by the parietal cells, but how does it secreted? Okay, so for that we have talked that this is the lumen of the gland, and here we have the this uh, one sodium potassium ATPase pump. Okay, and we have the sodium potassium ATPase pump, and here you can see yeah, sodium potassium ATPase pump. This is the blood side and this is the lumen side. Okay, so due to, and as the sodium so this potassium is again coming back by the potassium channel. Okay, and then also to maintain the gradient. Okay, to maintain the gradient, this H is also being uh, secreted outside from the parietal cells. And similarly here at the for the maintenance of the gradient for the bicarbonate ion and the so the bicarbonate ion is released outside, and in exchange of that another anion, which is chloride ion, is taken inside. Okay, so then this chloride ion is also being taken out from the other this what to say, a carrier receptor here. So this will also, this channel will throw the chloride and this one will throw the excess hydrogen ion. So these both together form the HCl. Okay, so here this second one is the sodium potassium ATPase pump here. Okay, and also this fourth one is an anion exchanger, which is this anion is going out and this chloride anion is coming in. Okay, and then we have this chloride exchanger, the third one. Okay, and here we have this. Okay, no, sorry, this one is the this second one is the uh, this hydrogen and potassium ATPase here. So here we are having the sodium potassium ATPase channel. Okay, so you can see sodium is thrown out and potassium is taken in, and that potassium whatever is taking is it taken out. Okay, and that again coming back with this sodium hydrogen potassium ATPase channel. So again potassium is coming in, hydrogen is taken out. Okay, so that is how it is happening. So here we are having this potassium and sodium potassium and hydrogen ATPase pump. Here we are having this sodium potassium ATPase pump. Here we are having this anion exchanger, and this one is having a chloride channel, and that's how the HCl secretion happens. Here this is the uh, picture that is showing us how the hormone is released. So this is you can say an endocrine cell, and these are the hormones inside it. These hormones are being released into the bloodstream. Okay, and then this hormones via the blood reaches to the target cells, which is having the receptor for the hormones. Okay, so this is our endocrine cell having the hormones is released in the bloodstream. This is the bloodstream. Then this is the from the bloodstream it goes to the target cell. Here you can see this is target cell, this fourth one is target cell, and the sorry third one is target cell, fourth one is our receptor for the hormones. And this is uh, the effect of the growth hormone. So this one here it will be the growth hormone. Okay, this what does it do? So the growth hormone has a direct effect. So this is talking about the direct effect. So this one is the growth hormone's direct effect. So here what it is showing, it is showing a lipolysis event. So fat is being lysed as a direct effect of the growth hormone. And second effect is on the liver. What happens from the liver, the IGF-1 is being secreted, which is insulin like growth factor, which helps in the lengthening of the bone, which is the indirect effect of the growth hormone. Okay, so here these are the how the somatotrophs or in the pituitary gland the growth hormone release is being regulated. So there are one is growth hormone releasing hormone, another one which is somatostatin. So growth hormone releasing hormone positively regulates the growth hormone production and secretion. So here it will be growth hormone releasing hormone, where it is bind to the G protein coupled receptor. So this G protein allows the adenylyl cyclase to convert the ATP to cyclic AMP. The cyclic AMP activates the protein kinases, which phosphodilates the CRAVE, and CRAVE enters and activates the transcription of FOS, FIT1 and growth hormone synthesis. Okay, growth hormone synthesis is increased and secretion of growth hormone is happening. Okay, proliferation that ultimately leads in the proliferation. Okay, and is this third one is our somatostatin, which also binds with the receptor, and that receptor inhibits the adenylyl cyclase and reduce the cyclic AMP production, that ultimately inhibits the growth hormone production. And this is a negative feedback loop. Okay, so this is an endocrine axis driven negative feedback. So here what we can say, so this is what here we are seeing the peripheral endocrine gland, which releases the hormone on the target cells, and physiological effects can be seen. So peripheral endocrine gland, so from where, so these peripheral endocrine glands are being stimulated by a tropic hormone. Okay, so this will be a tropic hormone, and tropic hormone is released from where? It release from the pituitary gland. Okay, and pituitary gland releases the tropic hormone under the influence of releasing hormone, and that releasing hormone is coming from hypothalamic neuroendocrine neurons. Okay, so that is how the loop has happened. So from neuroendocrine hypothalamic neurons, they release the releasing hormone which will act on the pituitary gland that releases the trophic hormone, and the trophic hormone acts on the peripheral endocrine glands that releases the final hormone that acts on the target cell and allow the physiological effects to take place. These hormone having the negative effect on the pituitary gland as well as the hypothalamic neuroendocrine neurons. Okay, so these are all the details of the endocrine system. If you have any doubt, you can ask me.
Currently, no, ma'am.
Okay. If you have anything to ask, you read and come to the next class, and then also you can ask the same doubts. Okay.
Yes, ma'am.
Okay. So see you in the next week. I'm concluding the session.
Thank you, ma'am.